And you haven't started anatomy yet, right? You didn't study anatomy this morning. But for sure you remember in your previous studies, the first thing that they teach is the anatomical position. They show you a person in the anatomical position, and they teach you anterior, posterior, superior, inferior, the cavities, etc. That's complicated, too many details, too many things to remember. In physiology, that's the human body. That's the physiological position. Okay, our body is an organism, or is a system, to be more physiologically accurate. We are a system that is enclosed, okay, by the skin, which is the barrier between us and the external environment. And then the inner part of the body is subdivided into two big compartments. One is the intracellular. If you take all the cells of the body and make one compartment, that is the intracellular compartment. And then we have the extracellular compartment that is subdivided into, one is the blood plasma, and the other is the interstitial fluid, that is the fluid that is bathing the cells. Okay, and what separates these compartments? We have more information in the next slide, so you don't need to worry now because of the lack of labels there. Between the cells and the interstitial fluid, there is a cell membrane. Okay, and between the blood and the interstitial fluid, we simply have the walls of the blood vessels. Okay, the capillary walls. Okay, that's a very important difference, as we are going to see later, with the cell membrane. This one here has more information. Okay, notice that we have on the right side the intracellular, on the left side we have the extracellular. Okay, that diagram has a lot of good information, but I don't like the proportions. Okay, because the intracellular there is represented as smaller than the extracellular, and that is not the case. Notice, you go back here, most of our body fluids, most of the water that we have in the body is inside the cells. Okay, 2/3 of the total body water is inside the cells. The other third is outside. Okay, that extracellular compartment, if you divide it, you can say that a quarter of it, 25%, is the plasma, and 75%, or 3/4, is the interstitial fluid. So, yeah, we are going to use information from the two diagrams. What do we have here? First of all, notice that in between the intracellular and extracellular compartment, we have this double layered barrier. That is the cell membrane. Probably you remember it's a bilayer of phospholipids. It's a highly selective membrane, okay, regarding permeability. Okay, what enters and goes out of the cell is very tightly regulated. And what separates the blood plasma from the rest of the extracellular fluids, the interstitial space, that is the capillary wall, the capillary endothelium. Okay, cells that sometimes they are very, very tightly joined together. For example, in the brain, okay, these endothelial cells have proteins that allow the attachment between one another. Some of these attachments are called tight junctions. In the case of the endothelial cells in the brain, there are 5 layers of tight junctions. So it is very difficult for anything to pass in between the cells. But if you go to the liver, if you go to the spleen, if you go to the bone marrow, these cells are actually a little bit more loose. There are gaps in between the cells. So it is easier for things to move in between the cells. What else is important from here? Let's take a look at the composition. Okay, we have the intracellular fluid. Notice that we have certain amount of sodium, potassium, chloride, and some proteins. Notice that the most abundant electrolyte in the intracellular fluid is potassium, 120 millimoles. There is a very little amount of sodium and chloride. And there are some proteins there. What happens in the extracellular fluid? Notice the amount of sodium, 145. Okay, so there is a lot of sodium outside the cells, very little inside the cells. What about potassium in the extracellular fluid? 4.5. Okay, compared to the amount of potassium inside the cells. There is a huge difference. We could say that most of the sodium is outside the cells and most of the potassium inside the cells. And who knows who's responsible for that? How do we guarantee that potassium is inside, sodium outside?
The sodium-potassium pump.
Sodium-potassium pump. And that sodium-potassium pump, how does the sodium-potassium pump does that?
It maintains. Okay. Go ahead.
No.
Through ATP.
Using ATP. Using ATP, and to make ATP what the cells need. How the cells produce ATP? There are different ways, but what is the most important? What is the organelle that produces ATP? Most of it. Mitochondria. What mitochondria do to make ATP? What they need to make ATP? They break down carbs, fatty acids, but they need, if you say burn, in order to burn, you need something. Oxygen. Okay, if you have a flame and you cover it, what will happen with the flame? Because there is no oxygen. Okay, if you need to burn something, there has to be oxygen. Okay, if there is a fire, one of the things that we can do is cover it, so there is no more oxygen, and that's it. Okay, so those are the reasons for which we eat and breathe. Mitochondria need glucose or fatty acids and oxygen to make ATP, and that ATP is going to be used by the sodium-potassium pump to take all the sodium out as they can and all the potassium back into the cells and maintain that difference. That difference in the concentration of potassium and sodium at both sides of the cell membrane is what we call the physiological steady state. Very important concept. And to maintain that steady state, ATP is needed, and that's why we have to go to the supermarket. And to go to the supermarket, you need money, and to make money, you need to work. Okay, oxygen, up to now, is free. There is no tax for breathing. So what happens if cells stop receiving oxygen? What will happen? Who can tell me? Prediction. What will happen if cells suddenly stop receiving oxygen? But be more specific. If a mitochondria doesn't get oxygen, what will happen? Proteins. Mitochondria make proteins. ATP won't be produced. And if there is no ATP in a cell, who will stop working? Yeah? Who was using ATP before? The sodium-potassium pump won't work. So what goes into the cells? Is someone going to take it out? And what happens with the potassium that moves out of the cells? Is someone taking it back? So when the sodium-potassium pump doesn't work, sodium will enter, because you know what is diffusion? Diffusion is when ions move following their concentration gradient from high to low. There is a moment when there is the same concentration of sodium outside and inside the cells, and that's it. Cells become salty, and potassium escapes, so that's not good. That is what we call reaching a chemical equilibrium, and equilibrium is not very good in that example. Okay, because if you remember, when sodium concentration increases in one place, what is the substance that follows sodium? Huh? Water. So now we have a cell that has more sodium and water. What will happen to that cell, to the size of the cell? Swell. And if that is happening in my skin, that is not a big deal. But if that is happening to my neurons, that is a problem. And that's why when we don't receive oxygen to the brain, for some minutes, the neurons stop working, and you know what happens. Don't have to tell you. So all that is the reason why we need to work, buy food, eat food, and continue breathing. Now, as you see there, there is a slight difference in the composition between the blood plasma and the interstitial fluid, but that is not important for us now. There is one thing that is important, okay, talking about the differences between interstitial fluid and the blood plasma, and it's the amount of protein. Notice that inside the blood, in the plasma, there is an important amount of protein. Okay, in the interstitial space, notice that the protein is, I don't like that 0. You can put 0.1 because there are proteins there. It's not 0. There are proteins, of course, but very little amount of proteins compared to the blood. And why is that? Well, remember the capillaries, okay, no matter how tight they are going to allow the pass of ions. Okay, little ions, sodium, potassium will move almost freely in and out, but proteins can't move. Okay, the little gaps between the endothelial cells are not big enough for proteins to move from the plasma into the interstitial space. Okay, so those are the basic differences between the composition of these fluids. Now, I want you to look at another value that we haven't mentioned, and that is osmolality. Osmolality is a measure of the concentration of all the solutes that are present in a solution. We have into account sodium, potassium, phosphate, chloride, urea, alcohol, sugar, everything that is present there. Okay, that gives us an idea of the osmolarity. Okay, in the blood plasma, notice that the osmolality is 290. In the interstitial fluid is same, and in the intracellular compartment is the same. All the compartments of the body have exactly the same concentration of solutes. Those solutes are different, but they have equal concentration. Okay? And that's important because osmolality is what drives the movement of water. Okay, so for example, let's say we administer to this person a solution that contains a high amount of salt. Now we are increasing the osmolality of the plasma. Okay, what will that determine? That water will move from the interstitial space towards the blood. When water moves from here to there, now the osmolality increases here. And what will that determine? That water will move from the cells to the interstitial space. And the cells are going to shrink. So don't drink seawater, because that is what will happen. On the contrary, if we administer distilled water, pure water, now we are decreasing the osmolality here, and that will make the water go here and then there. Okay? So imagine you have a person who has lost 1 liter of blood, and you have an IV catheter, but the only thing you have is distilled water. Will that help increase the blood pressure? If they lost 1 liter of blood, and you put 1 liter of pure water in the blood, what will happen with that water? So imagine that the liter of water will distribute in the entire thing. How much is going to be left in the blood? Almost nothing. So that is not going to increase the blood pressure. For that reason, we use normal saline, which is isotonic, has the same osmolality of the blood and the body. So we guarantee that the liter stays in the blood vessels. Now, here you have a table that is about the composition, and before anyone gets scared, in physiology and pathophysiology, maybe one day I will tell you, I need you to remember this figure. I will not normally ask you for any of these figures. You're going to find questions of the type, which of the following has the greatest, smallest, but not numbers. Let's give you the example of the amount of sodium. Okay, the plasma has 142. Okay, we have protein-free plasma, interstitial cells. We don't need the one in the center. But let's take the plasma, interstitial, and the cells. 142, 145, 15. Imagine that someone asks you a question. Which of the following is the concentration of sodium in the interstitial space? And they give you A, 142, B, 143, C, 144, D, 145. Oh my goodness, I don't remember exactly. I remember one was 142, 145, but I don't remember which. I'm not going to ask you that. Which of the following compartments has the highest sodium concentration? Okay, and it will be extracellular compartment, intracellular compartment, intramitochondrial compartment, something like that. Okay, so you remember that sodium goes out of the cells, potassium inside the cells. That's the concept that I need you to remember.